I want you to just imagine a highly specific clinical scenario for a moment.
Okay.
You've got a patient sitting in an oncology exam room, and they have recently been diagnosed with pancreatic cancer.
Right.
But when you look at the imaging, the tumor itself is actually, quite small. Visually on the scans, it hasn't formed this massive bulk that's crushing adjacent organs or anything.
It's localized.
Exactly. Yet this patient is experiencing intense, debilitating, just radiating pain that seems, frankly, completely out of proportion to the physical size of that mass.
Yeah.
You look at the scans, you look at the patient in total agony, and the physics of it just do not make sense. Why is a localized, relatively small tumor causing systemic-level agony?
I mean, it is one of the most agonizing and historically frustrating paradoxes in oncology.
Yeah.
And that the reality is the mechanics of that pain, they just don't make sense until you stop looking at the tumor mass as a singular object.
Okay, so what should we be looking at?
You have to start looking at the microscopic wiring surrounding it. What you're describing is driven by a phenomenon known as peritumoral invasion, or PNI.
PNI. Got it. So is that just like the tumor growing and bumping into a nerve?
No, not at all. This isn't just applying pressure. PNI is an active, really aggressive process. Cancer cells physically crawl along nerves, they invade the protective nerve sheaths, and they completely hijack the local neural circuitry.
Wow. And, deciphering exactly how that hijacking happens, that is the core mission of today's deep dive.
Absolutely.
We are working through a pretty dense stack of source material today, centered mostly around a really groundbreaking 2026 paper published in the journal Neuron.
Yeah, by Zhang and colleagues.
Right. And our goal today is to basically decode this research entirely for you. We are going to look at the cellular players, the molecular signals, and the dark biological machinery that creates this incredibly toxic relationship between pancreatic cancer and your nervous system.
What's fascinating here is the paradigm shift introduced by Zhang and this team. It's really profound.
How so?
Well, for decades, the dominant approach in oncology was to treat a solid tumor like an isolated fortress of rogue cells, right? You attack the fortress.
Right.
But this research proves that the problem doesn't initiate solely within the tumor, and it doesn't start in the nerve itself either.
Okay, then where does it start?
The epicenter of this agonizing invasion is actually in the biological neighborhood surrounding the tumor. We clinically refer to it as the peritumoral microenvironment.
The peritumoral neighborhood. Okay, so to understand the crime, we really have to understand the neighborhood where it takes place.
Exactly.
Let's look at the anatomy of pancreatic ductal adenocarcinoma, or PDAC. The bulk of a healthy pancreas, it isn't actually made of duct cells, right? It's made up of acinar cells.
Yeah, acinar cells.
And the word acinus, it actually comes from Latin for berry, because under a microscope, these cells cluster together like little bunches of grapes.
That's right.
And their day-to-day job is just exocrine function. They are the local factories producing the digestive enzymes that ultimately help you break down your food.
Right, they are highly specialized, very stable cells under normal conditions.
But things change when cancer shows up.
Drastically. When pancreatic cancer begins to develop, we see the emergence of early precursor lesions. These are termed PANIN, which stands for pancreatic intraepithelial neoplasia.
Okay, PANIN.
Yeah. And as these microscopic lesions form, a massive disruption occurs in the surrounding tissue. Those healthy acinar cells begin to undergo a process called ADM.
Which is acinar to ductal metaplasia.
Exactly.
I actually want to pause on that term, metaplasia, because it's pretty crucial here. We aren't talking about these acinar cells mutating into cancer themselves, right?
Correct. No, metaplasia is actually an evolutionary defense mechanism.
Oh, really?
Yeah. When these acinar cells are subjected to intense inflammatory stress from the neighboring early-stage tumor, they sort of shed their original identity.
Wow, like a cellular identity crisis.
That's exactly what it is.
Yeah.
They stop functioning as enzyme-producing berries, and they physically transform into duct-like cells just to survive the hostile environment.
Okay.
So in this peritumoral zone, you now have a massive population of deeply stressed transforming cells.
But wait, I need to challenge the sequence of events a bit. Even if these acinar cells are stressed and having this identity crisis, they are still fundamentally native, non-cancerous cells.
Yes.
So why do these innocent neighbors suddenly become active tumor helpers? What flips that switch?
That is the pivotal question. And the answer lies in a tragic biological misfire by the patient's own immune system.
The immune system.
Yeah. The researchers meticulously mapped the immune cells patrolling this neighborhood, specifically isolating CD4 plus T cells.
Okay, CD4 plus T cells, those are usually the good guys, right?
Exactly. They're typically the conductors of your immune orchestra. They coordinate attacks against infections and abnormal cells.
Right.
But in this specific tumor microenvironment, these T cells behave aberrantly. They begin continuously releasing a specific cytokine, a signaling protein called interleukin-22 or IL-22.
So the immune system is actively sending a signal, IL-22, right into the stressed neighborhood.
Yes.
What happens when that IL-22 signal lands?
Well, IL-22 floats through the extracellular space and binds to a highly specific receptor called IL-22RA1. And that receptor is located directly on the surface of those stressed acinar cells.
Okay, let's unpack this. What does that binding actually do?
When that receptor is tripped, it forces the acinar cell into overdrive. The cell essentially abandons its normal biological programming and starts mass-producing a family of proteins called Reg proteins.
Reg proteins.
Specifically, regenerating islet-derived protein 3A, or Reg 3A.
Okay, think about the mechanics of this for a second. The CD4 plus T cells are basically acting like a corrupted central command, right? They're issuing this emergency broadcast via IL-22.
Right.
And the acinar cells, they receive this order, they shut down their normal enzyme production, and they become biological printing presses.
That's a great analogy.
They're churning out thousands of these Reg 3A proteins, these sort of malicious love letters, and they don't just keep them internally, right?
No, not at all. They utilize paracrine signaling. They are actively pumping these proteins out into the surrounding neighborhood, thoroughly saturating the extracellular space.
Wow.
And the density of these red proteins in the tissue is staggering.
Yeah.
To your earlier point, the tumor itself isn't manufacturing these proteins.
Right, it's the neighbors.
Exactly. The non-cancerous, structurally intact surrounding tissue is actively driving the pathology here.
So if we just do a quick recap, kind of synthesize what we've covered in these first 5 minutes or so, we are looking at a deeply concerning chain reaction.
Yeah.
Pancreatic cancer drives severe nerve invasion and intense pain, but the primary fuel for this process originates from the tumor's neighbors.
Yes.
The immune system's T cells mistakenly release IL-22, which then forces the acinar cells to flood the microenvironment with Reg proteins.
That is the exact circuit.
So I pose this question to you, the listener. Think about the evolutionary imperative of your own immune system. Isn't it terrifying that the very T cells designed to protect you are actually the ones pulling a biological fire alarm that inadvertently arms the cancer?
It is deeply unsettling, yeah.
And if this peritumoral space is practically drowning in Reg proteins, the next logical mystery is, who is catching those signals?
Because a signaling protein floating in the void is entirely inert unless it finds a compatible receptor.
Exactly.
The researchers had to identify what structural component was actually receiving these Reg proteins. And they discovered that the functional receptor is a protein called EXTL3.
EXTL3.
Yeah, it stands for exostosin-like glycosyltransferase 3.
Okay, that is a dense biochemical name. Let's break down its normal function versus its role here.
Sure. Under standard physiological conditions, EXTL3 is an enzyme involved in synthesizing complex sugar chains, things like heparin sulfate, within the cell. However, the researchers discovered that on the surface membranes of specific nerve cells and cancer cells, EXTL3 acts as a direct docking station.
Oh, like a mailbox.
Exactly like a mailbox. It functions as the physical receptor that catches and binds the Reg 3A proteins.
Let's follow the signal to the nerves first, because we really have to address that initial clinical paradox we talked about, the excruciating, completely disproportionate pain.
Right.
The source material specifically zeroes in on a class of sensory nerves called TRPV1 plus nociceptor.
Yes, nociceptors are your body's primary biological pain alarms.
Okay.
And the TRPV1 receptor is fundamentally critical to how we experience severe pain. To give you a real-world equivalent, TRPV1 is the exact same sensory receptor that is triggered by capsaicin.
Wait, like from chili peppers?
Exactly. The active chemical compound that makes chili peppers feel hot and physically painful. That's TRPV1.
Oh, wow. So when the Reg 3A protein binds to the EXTL3 receptor on these pain nerves, is it acting like capsaicin? Does it just force the nerve to fire a pain signal?
Actually, no. The mechanism is far more insidious than a direct trigger.
Really?
Yeah. The research team isolated these specific TRPV1 neurons and directly applied Reg 3A to them. They observed that Reg 3A does not directly force the calcium channels to open.
Okay.
It doesn't initiate the electrical action potential that the brain registers as pain. Instead, It potentiates the neuron's response to other ambient stimuli.
Oh, I see. I want to make sure this physiological nuance is crystal clear for you listening. Think of the nerve's baseline sensitivity like a finely tuned microphone, right?
Yes.
And the physical pressure from the growing tumor is the ambient sound in the room. Reg3A doesn't make a sound itself.
Right.
What it does upon binding to EXTL3 is alter the internal gain on that microphone. It radically lowers the activation threshold of the nerve. So a minor fluctuation in pressure, something that should be completely painless, suddenly floods the neuron with calcium.
Exactly. It registers as a deafening, agonizing roar. It induces extreme mechanical hypersensitivity.
That is awful.
And the pathology of Reg3A on the nervous system extends beyond just hypersensitivity.
There's more?
Oh, yeah. These proteins possess potent neurotrophic properties, neuro referring to the nerve and trophic indicating growth or nourishment.
Okay, so they make the nerves grow.
Yes. When Reg3A continually binds to the EXTL3 receptor, it forces the nerve to structurally alter itself. It triggers neurite outgrowth.
Wow.
The nerves physically sprout new branches, and they extend rapidly toward the highest concentration of Reg proteins, which happens to be the tumor itself. We clinically define this as hyperinnervation.
I mean, it reads almost like a dark science fiction narrative, but the researchers proved this mechanism definitively in vivo, right?
They did. They utilized KPC mice.
And for context for the listeners, the KPC mouse is basically considered the absolute gold standard animal model in pancreatic cancer research.
Yes.
Because its genetic mutations perfectly mirror the aggressive progression of human pancreatic ductal adenocarcinoma.
Exactly. So the research team took these specialized mice and genetically engineered them to knock out the XTL3 receptor exclusively in the TRPV1 pain nerves.
And what happened?
The resulting data was remarkably definitive.
Yeah.
By removing that specific EXTL3 docking station from the nerves, the physical sprouting, the sensory hyperinnervation was dramatically halted.
Wow.
And more critically, the mechanical hypersensitivity, which is how we measure pain behaviors in the animal model, it just plummeted.
That's incredible.
The surrounding environment was still flooded with Reg proteins, but the nerves were rendered functionally blind to them.
So let's take a quick pause here for our second recap. Look at the board right now. We've uncovered a massive piece of the puzzle. EXTL3 is the specific receptor catching the Reg proteins.
Yes.
On pain neurons, this interaction doesn't cause pain directly. It fundamentally lowers the pain threshold, making the nerves hyperreactive. And simultaneously, it acts as a biological fertilizer, forcing a dense web of pain nerves to grow directly toward the cancer.
It's a perfect summary.
So, a strategic question for you to ponder. If you were designing a drug right now, would you try to block the Reg proteins floating in the space between cells, or block the EXTL3 receptor on the nerve itself?
It's a complex problem.
It is.
Yeah.
Because the nervous system is only half of this equation.
Exactly. Here's where it gets really interesting. We've established that the nerves are hypersensitive and physically drawn to the tumor, but the cancer cells are not passive entities just waiting to be surrounded.
Right.
The cancer has to physically mobilize to invade those newly grown nerve sheaths.
So if the nerves are building the highway, how does the cancer build the vehicle to drive on it?
It is driven by the exact same peritumoral Reg proteins we've been tracking.
The cancer cells have the EXTL3 mailbox, too.
Yes, the PDAC cancer cells prominently express EXTL3 receptors on their own surface membranes. And when Reg3A binds to the cancer cell's EXTL3, it initiates a completely different, highly complex intracellular cascade.
Okay, let's deep dive into this mechanism.
It's basically a molecular domino effect that travels from the outer membrane all the way straight into the nucleus.
And the researchers meticulously mapped this cascade. Understanding the why behind these chemical steps is crucial. So step 1, Reg3A docks at the EXTL3 receptor on the exterior of the cancer cell. What happens directly inside the membrane?
The internal domain of the EXTL3 receptor undergoes a conformational shift, and it physically binds to an intracellular protein called XIAP.
XIAP. Okay. Historically, isn't XIAP recognized by oncologists as an X-linked inhibitor of apoptosis?
Yes, meaning its primary job is normally to prevent the cell from undergoing programmed death.
Right.
But in this specific context, XIAP is moonlighting. It's functioning purely as an adapter protein, a bridge to the next system.
And the research is extremely rigorous here. The paper notes they also observed EXTL3 binding to another protein called ATXN1.
Yes, they did.
But through rigorous knockdown experiments, they proved that only the XIAP pathway actively drives the invasion mechanism.
Correct. Only XIAP.
So XIAP is the key. Where does this bridge lead?
SIEP interacts with and hijacks a massive lipid-regulating complex known as the INSEG-1-SCAP SREBP1 pathway.
Okay, wow, that is an absolute alphabet soup right there.
Right, yeah, no, it's dense. But the critical output you need to focus on is SREBP1.
SREBP1, got it.
Exactly. SREBP1 is a transcription factor. Under normal circumstances, it's tethered outside the nucleus, regulating cellular cholesterol and lipids.
Okay.
But the SREP interaction triggers an enzymatic cleavage. SREBP1 is physically cut loose from its tether.
Oh, wow. And once a transcription factor is cut loose, its singular goal is to alter genetic expression.
Yes.
It dives straight into the nucleus of the cancer cell, binds to specific target genes within the DNA, and initiates a massive reprogramming event.
Exactly.
But why does a cancer cell need to hijack a lipid-regulating pathway to invade nerves?
Because to move, the cell must completely rebuild its physical structure, specifically its lipid membrane.
Ah, that makes sense.
And the culmination of this genetic reprogramming is the explosive cellular synthesis and secretion of TGF-beta, transforming growth factor beta.
Okay.
The cancer cell begins pumping massive quantities of TGF-beta into its immediate vicinity, creating what we call an autocrine loop.
Which means the cancer cell is feeding itself, basically.
Right. The cell secretes the factor, which then binds back onto receptors on that exact same cell, continuously amplifying the signal.
And TGF-beta is the master architect of a process called EMT, right? Epithelial mesenchymal transition.
Yes, EMT.
To visualize how destructive EMT is, think of normal, healthy epithelial cells like a tightly mortared Lego castle.
Good analogy.
They are structurally rigid, securely locked together to their neighbors. They cannot mobilize.
Right.
TGF-beta acts as a highly potent chemical heat ray that literally melts the Legos. It downregulates the adhesion molecules, reverting those rigid epithelial cells into a fluid, highly mobile, slime-like state.
That's exactly right. Once that structural integrity is dissolved, the cells are essentially untethered. They can detach from the primary tumor mass, physically squeeze through the extracellular matrix, and utilize those newly grown hypersensitive nerves as biological highways.
And that's the PNI.
Yes, crawling along the neural sheaths to disseminate and metastasize.
So let's bring it all together for recap number 3. We now know the red proteins strike both sides of the equation.
Yep.
On the cancer cell, binding to EXTL3 triggers an internal domino rally. XIAP acts as a bridge to cut loose SREBP1, which travels to the DNA to mass-produce TGF-beta.
Exactly.
That TGF-beta causes EMT, melting the cancer cells into a mobile state so they can crawl along the nerves. We now have a complete circuit. Acinar cells signal the nerves to grow and the cancer to move. It's a perfectly orchestrated biological heist.
It really is, entirely orchestrated by the stress signals originating from the neighboring tissue.
But decoding the circuit is only half the battle. The ultimate question for you, the listener, is how do we stop it?
Right. How do we break the cycle? Translating this from a molecular map to a viable therapy is where the true impact of this research lies.
So what did they do?
Well, the investigators didn't stop at merely observing the pathway. They actively attempted to dismantle it pharmacologically in the KPC mouse models.
Okay, so targeting that specific intracellular domino rally.
Yes. They utilized 2 distinct pharmacological agents. They administered a drug called TL32711, which is specifically designed to inhibit the adapter protein XIAP.
Okay, TL32711 for XIAP.
And simultaneously, in separate cohorts, they used a compound called fatostatin, which is a known inhibitor of the SREBP1 activation process.
Got it. So if you block those internal steps, what happens to the systemic disease?
The pharmacological blockade was remarkably effective. By inhibiting either SIAP or SREBP1, the cancer cells were genetically blocked from producing that critical surge of TGF-beta.
So the Lego castle didn't melt.
Exactly. The cellular mortar was not dissolved. The researchers observed a massive reduction in EMT markers. The cancer cells remained rigid and anchored.
That is incredible.
Consequently, they saw profound reductions in metastasis, specifically the spread to the liver and lungs, which is typically fatal in pancreatic cancer.
And what about the pain?
Critically, because the interaction with the nerves was disrupted, they recorded significantly reduced pain behaviors in the subjects.
That is exceptionally strong preclinical evidence. But, as we know, proving a concept in a genetically accurate mouse model is a massive hurdle, but human biology is notoriously complex.
It is.
Mouse models do not always perfectly mimic human pathology. We really need to level up the evidence here. Does this intricate molecular dance actually translate to human beings currently fighting for their lives in oncology wards?
It absolutely does.
Yeah.
And the clinical correlative data in this paper is incredibly robust.
Oh, really?
Yeah. The research team analyzed tissue samples and comprehensive clinical data from a cohort of 205 human pancreatic cancer patients.
105, okay.
They were specifically looking for a correlation between physical nerve invasion and the molecular markers we've been discussing.
Yeah.
And they discovered an undeniably strong direct correlation.
They identified what we can essentially call a molecular triple threat signature in human pathology, right?
Yeah.
Let's detail what that looks like under the microscope.
When pathologists examined the tissue of patients who presented with severe perineural invasion, they consistently found 3 aligned factors.
Okay, what are they?
1, extraordinarily high concentrations of reg proteins saturating the peritumoral neighborhood.
Right.
2, highly elevated expression of the EXTL3 docking station on the tumor cells themselves.
Makes sense.
And 3, massive local concentrations of TGF-beta.
So if a patient's postoperative pathology report returned showing that exact triple threat signature, what was the reality of their clinical outcome?
The clinical reality was exceptionally grim. Patients actively expressing this complete signaling pathway had statistically significant decreases in overall survival rates.
Wow.
The disease was simply more aggressive and more prone to rapid metastasis. But equally important to survival was the metric of their day-to-day suffering.
The pain.
Yes. The researchers cross-referenced this molecular signature with the patient's reported pre-surgery pain scores.
And the stratification there is really telling.
Very telling. Patients whose tumors did not express this pathway generally clustered in grade, mild manageable pain. But the patients harboring this triple threat were overwhelmingly clustered in grade 2, reporting moderate to severe intractable pain that heavily degraded their quality of life long before surgery was even an option.
If we synthesize the broader implications of these findings, targeting this Reg-EXTL3 signaling axis represents something akin to a holy grail in pancreatic cancer pharmacology.
If we connect this to the bigger picture, absolutely.
Because the historical difficulty in treating PDA is that we are constantly chasing the metastasis while trying to manage the agony, right?
Right. But this pathway offers a theoretical 2-for-1 therapeutic benefit.
Exactly. Because if pharmacologists can successfully develop a targeted agent, maybe a monoclonal antibody that neutralizes Reg3A in the extracellular space, or a small molecule inhibitor that locks down the EXTL3 receptor, we aren't just treating one symptom.
No, we could simultaneously halt the physical structural changes that allow the tumor to metastasize, while also cutting off the molecular signal that plunges the patient's nervous system into a state of hyperreactive agony.
We could theoretically arrest the disease progression and the debilitating pain with a single pharmacological strike.
It forces a fundamental reevaluation of our therapeutic targets.
Oh.
We have proved that the tumor is not an isolated, self-sustaining entity. It is a highly sophisticated manipulator of its microenvironment.
We have navigated a massive, incredibly dense biological landscape today. Let's pull all the threads together for a final view of the board.
Let's do it.
The pathogenesis of this agonizing invasion doesn't begin with a cancer cell mutating. It begins with an immune error. T cells mistakenly flood the microenvironment with IL-22.
Right.
This inflammatory stress forces the innocent neighboring acinar cells to abandon their normal digestive functions and pump out massive quantities of Reg3A proteins.
Yep.
Those Reg proteins find the EXTL3 receptors on local pain nerves, radically lowering their activation threshold, turning up the volume on the pain signal and forcing them to sprout directly toward the tumor. Simultaneously, those exact same Reg proteins dock at the EXTL3 receptors on the cancer cells, initiating an internal cascade through XIAP and SREBP1 that ultimately unleashes TGF-beta.
And that melts Legos.
Yes, that chemical solvent dissolves the cellular mortar, allowing the cancer to melt into a mobile state and invade those newly grown hypersensitive nerves. It is an absolute masterpiece of dark, synchronized biology.
It really is a stark reminder of the complexity of the environments we're trying to treat. The cancer leverages the host's own cellular defense and repair mechanisms against them.
And that leaves us with a final provocative thought to carry with you. Historically, for over a century, the philosophy of oncology has been relentlessly tumor-centric.
Right, attack the tumor.
The protocol is always to attack the rogue mass, excise it with surgery, incinerate it with targeted radiation, or poison the rapidly dividing cells with systemic chemotherapy.
Yeah.
But the deep dive into this source material reveals that the tumor is fundamentally reliant on manipulating its healthy, innocent neighbors to execute its most lethal and agonizing behaviors.
Which changes everything.
It does. So I will leave you with this to ponder. What if the future paradigm of curing highly lethal diseases like pancreatic cancer isn't focused on designing more toxic weapons to kill the cancer cells directly?
Right.
What if the key to survival is simply cutting the biochemical phone lines to the neighborhood, preventing the cancer from ever issuing the orders in the first place? Think about how this concept of the microenvironment might apply to other instances of chronic inflammation, autoimmune disorders, or ailments within your own body.
It's a profound thought.
Are the rogue cells the true source of the problem, or is it the cellular environment that is passively enabling them to thrive? Keep questioning the underlying mechanisms. Until next time, keep diving deep.